One important trait to understand is the process of how to read a pedigree, which is a chart or family tree that tracks which members of a family have a particular trait. And we've seen this already, but now let's learn how to read it. A circle will always represent a female. A square will always represent a male. When there is a horizontal line connecting a male and a female, that represents a marriage, as you can see here. So this female and this male are married. A vertical line and a bracket will connect parents to their children. So we know that these individuals here are the offspring of these two parents. A square that is completely shaded or a circle that is completely shaded will indicate a person who has a particular trait. So here you can see this individual has a trait, this individual does not. A half-shaded circle or square indicates a person who is a carrier of a trait. For example, this person has a specific trait in its genome, so in its genotype, but not necessarily in its phenotype. You don't actually see it expressed. A circle or square that is not shaded in indicates that the person does not have the trait, nor is that person a carrier of that trait. One thing that scientists have been able to do is they have been able to develop the technology to create something called a karyotype, which is simply a picture of all of the chromosomes in a cell. And this image here is an example of a karyotype of the human chromosomes. You can see here that they are grouped in pairs, and you can see that each of the chromosome pairs looks a little bit different. Remember that each chromosome has different genes on it. Selective breeding is an interesting process of selecting organisms with desired traits to be parents of the next generation. I'll show you an example of selective breeding in a moment. Inbreeding is the crossing of two individuals that have very similar characteristics. Because they are genetically very similar, they may inherit alleles that ultimately lead to genetic disorders. And when we look at the breeding of certain animals, like dogs or horses, you might notice that there are some genetic disorders, difficulty breathing, hip issues in certain dog varieties, and that is because of the genetic similarity. Hybridization crosses two genetically different individuals. Cloning is an organism that has exactly the same genes as the organism from which it was produced. And you will have an opportunity to research one famous clone, Dolly the sheep. The reason why this individual has the same genes as the organism from which it was produced is because they actually use the nucleus of that original organism to produce the new animal. Here is an example of selective breeding, sometimes called artificial selection, because farmers and breeders only allow plants and animals with desirable traits or characteristics to reproduce. This is an example of how, over time, farmers have used the wild mustard plant to create all of these vegetables. So here you can see just some examples. Kohlrabi here came from a wild mustard plant because of an enhancement in the stems. Kale came from wild mustard because it enlarged the leaves. So they selectively bred wild mustard plants in order to ultimately create kale. Broccoli comes from wild mustard because they were able to suppress flower development. Brussels sprouts also come from wild mustard because of a selective breeding that looked at lateral leaf buds. So you can see that that ultimately leads here. Cabbage comes from wild mustard because of the suppression of the terminal leaf bud. And cauliflower, also through selective breeding, comes because of the sterility of the flowers. So there's a modified trait in the flower buds. Finally, as scientists have learned more about genetics, scientists have been able to begin doing something called genetic engineering. In this process, genes from one organism are transferred into the DNA of another organism. We discussed this very briefly when discussing the light sensitivity that scientists are using to affect anxiety, and they've tested this in rats. Some examples of how scientists are able to do this include through bacteria. An example of this is insulin. Because bacteria reproduce very quickly, large amounts of insulin can actually be produced in a very short amount of time. Other organisms can be used. For example, cells of plants such as tomatoes and rice are able to survive really cold temperatures and resist insects such as different pests. Finally, gene therapy is the process actually of inserting copies of a gene directly into a person's cells. The question is, are plants or agriculture safe to eat if it has undergone gene therapy? Finally, a further area for research that you will have is the opportunity to examine the Human Genome Project. The genome refers to all of the DNA in one cell of an organism. The Human Genome Project has identified that there are at least 30,000 genes in the human body, and the average gene has about 3,000 nucleic acids or nitrogen bases in it. So this is a very long process, but an interesting one to do some further research on that you will have the opportunity to do very soon.